main purpose of this e-learning course on cancer drug discovery and development is to give you an overview of the discovery of drugs in terms of starting from idea to the discovery of the process and then preclinical development and clinical development for a student in the post-graduation or in the PhD courses with the background in biology, chemistry and pharmacy backgrounds. And we have taken typical care in terms of identifying the expert faculty from the industry, from the academy and from the clinicians. Basic scientists, industrialists and clinicians are involved in offering this course. And so, if you just to give an overview of the drug discovery and development, it has 4 different phases: the discovery part, the preclinical development, clinical development and post-approval monitoring. So, I will give you an overview of this discovery and development of the drugs. So, that it has the entire process of drug discovery and development takes anywhere 10 to 15 years, starting from the discovery in terms of target identification and its validation, and against this target screening of compounds to identify a hit and optimizing the hit into a lead molecule and lead optimization, and followed by the candidate selection and taking of the preclinical trials to the later part is the clinical trials, and the total costs that are involved will be somewhere around $1 billion US. So, the most important aspect of the drug discovery and development is identifying a target. If you see the various drug targets that are there in the market, most of the drugs were developed against the receptors. Receptors are the ones which are involved in receiving the signal and then transmitting to the internal cellular system for the effective responses. So, in response to any kind of stimulus, the receptors get activated and then execute the functions. Then followed by enzymes. Enzymes, the variety of enzymes have been exploited for the development of the drugs, followed by hormones, then DNA based enzymes and nuclear receptors, ion channels, and these are the targets which have exploited, out of which receptors, enzymes and hormones, most of them are protein in nature. And understanding the protein structure and function is very critical for development of a drug. And so, the drug discovery process in the past and present, if you compare, in the past we have been using the drugs were developed either by trial and error or by serendipitous discoveries. Today, efforts are made to understand the molecular basis of different diseases, and then you exploit them for designing and developing the specific drugs. In modern drug discovery process, bioassay screenings play an important role. So, the drug discovery process, choose a disease, identify the target, exploiting the tools that are there, genomics, proteomics and metabolomics, develop a bioassay against the target that was identified in terms of in vitro and in vitro either in test tubes or in cell cultures, and in vivo in animal models. And once you have a lead compound against this one, conventional screening you can do, or high throughput screening, or you can use the computer aided drug design approaches, CAD approaches or in silico approaches. Once these are done, the toxicity evaluation can be done through pharmacokinetics and dynamics and the other toxicity profiles. This discovery process, identifying the disease target is most critical, and developing a bioassay is another component. So, this identifying the drug target, it has to be specific. As I said, it could be a receptor, it could be an enzyme, it could be a biological system responsible for the disease manifestation. For example, cyclooxygenase 2 has been identified as the target for developing anti-inflammatory drugs. Inflammation is a critical component for against injury or infection, and that is mainly led by one of the metabolites of cyclooxygenase 2 enzyme, and cyclooxygenase 2 has become a big target for the development of anti-inflammatory drugs. Similarly, chronic myeloid leukemia is another one of the types of cancers in which the activation of the BCR-ABL kinase is responsible. The kinase has been identified as the target, and you exploiting the structure function correlation or three-dimensional structure of this BCR-ABL kinase, the drugs have been developed, and today in the market, the Gleevec that is present in the market for the treatment of chronic myeloid leukemia is developed against this target. So, choosing the right target is important. Otherwise, if you are not using the right target, then it may lead to all types of side effects. So, in this, as I said, selectivity is important. And for example, if you are trying in infectious diseases, when you are targeting an enzyme, the enzyme specific to the bacterial source is important. If the same enzyme is present in the bacteria as well as in the host, then it may lead to the side effects. So, the selectivity is important in order to prevent the side effects. And because of lack of this selectivity is responsible for many of the side effects that we see with the drugs that are present in the market. For example, if you take non-steroidal anti-inflammatory drugs, they are having the conventional non-steroidal anti-inflammatory drugs have gastric side effects or cardiac side effects, and this is mainly because of lack of selectivity. So, selectivity of the target is very important. So, once you have identified, understood the disease process, identified the right target, then you have to go for choosing the bioassay process. How do you assay against the target that you have imposed? These assays could be in vitro, that is an isolated enzyme systems, or receptors, or cells in terms of the cells in the culture media. And once you complete your screening process against the isolated enzyme or then cell lines, then you go for in vivo studies in animal systems, referring to specific disease animal models. If you have a right disease animal model, then you can see the efficacy of the drug in the animal models. In addition to this, ex vivo also you can take out the tissues and see the efficacy of the drug on the isolated tissues, which you call it as ex vivo assay. So, using against the target that you have identified, against the bioassay that you have developed, you screen the compounds to identify a lead compound. Lead compound is the one structure that has some activity against the chosen target, but it is not yet the final drug itself. Using this lead compound, you go for developing further molecule. This kind of lead molecule, it could be a synthetic molecule, it could be a natural product. In fact, the plants, microbes, marine world and the animals all provide a rich source of structurally complex and diverse molecules which could be exploited as against the targets that we have in mind. And the screening process could be manual or it could be high throughput screening approaches. And So, once you have identified through in vitro assays and isolated enzymes or cell lines and come up with a lead molecule, then you have to evaluate the efficacy of this molecule in the animal models, efficacy as well as safety. Usually the animal models that are employed are mice, rats, rabbits, guinea pigs, hamsters, and in some cases the primate animal models also. And so the most important thing in the preclinical trials, specifically in vivo studies, is determining the pharmacodynamics and pharmacokinetics of the drug. Pharmacodynamics explores what a drug does to the body. Once the drug enters into the body, what exactly it is doing to the tissues. Is it causing a toxicity to the liver or the kidney or the heart? And then the pharmacokinetics actually explores what the body does to the drug. Once the drug enters into the body, what exactly it is being undergoing transformations, because liver is the center of detoxification. As the drug enters into the body, the liver metabolizes the drugs and then eliminates from the body through kidney. And in this process, there are various detoxification procedures. The phase 1 detoxification involving cytochrome P450 systems, and phase 2 drug detoxification systems include glutathione transferases and other glucuronidases, example like that. The entire process of this drug detoxification, which we study under pharmacokinetics, is to eliminate the drug from the body. Basing on this, the time that it has taken, the dose interval and the dosages are determined. So, the pharmacokinetics and pharmacodynamics are very important in the thing. And as I said, the metabolism of the drug, as the drug enters into the body, then the body considers this as a foreign substance and then is metabolized, and this metabolism involves the phase 1 and phase 2 drug detoxification systems, which eventually eliminates this drug from the body through urine and other feces kind of thing. And in this, the liver is the most important organ of metabolism of the drugs, and the detoxification takes place. So, once you have completed the entire process of preclinical, so identifying the target, developing a bioassay process, the screening, identifying the hit molecule, the lead molecule, in vitro and in vivo assays are being done. Then before you proceed to the clinical trials, you have to design a manufacturing process and manufacture this under GMP conditions, and in order to carry out the clinical trials in human subjects. So, before you take up these clinical trials, you have to develop a process for synthesizing this drug molecule under good manufacturing practices laboratory. So, the entire clinical trials involves phase 1, phase 2, phase 3 and phase 4. Phase 1 is actually done on healthy volunteers to determine the toxicity of a relative to dose and to screen for unexpected side effects. So, a few number of healthy volunteers are taken and see whether it has any toxicity, and also how much time it is taking for elimination from the body. That is, pharmacokinetics and pharmacodynamics are done on healthy volunteers, and this in turn leads to determination of the dosage, and the dose interval will be determined in the phase 1. And phase 2 is actually conducted on group of patients, a small number of patients, to see if the drug has any beneficial effect and to determine the dose level that is required in order to get this one. So, efficacy of the drug is tested in phase 2 on a small group of individuals, and if it is found efficacious and without any kind of side effects, then it will be approval is given to enter into the large scale clinical trials, which you call it as phase 3. Here the drug is tested in much larger group of patients and compared with the existing treatments and with a placebo. And the placebo is a double blinded kind of trials that they conduct in order to see whether it is the beneficial effect that you see or because of the placebo or because of the drug that is. So, here multi-centered clinical trials are done on large number of patients and in order to see the efficacy and safety of the drug. Once this phase 3, phase 1, phase 2, phase 3 are completed, and if the results are showing that the drug candidate is safe and it is effective in curing the disease and it is much better than the existing drugs, approval is given by the drug controller authority for large scale marketing and distribution of the drugs. And even after the drug gets approval for marketing, still the trials will be conducted, which you call it as the phase 4 clinical trials, that is called as post market surveillance, in order to see the long term effects of the drug. See, the drug, all these clinical trials are conducted for a small number of individuals and also for a limited period of time. And if the drug is taken for longer periods, like for example arthritis, the drug has to be taken lifelong, or cardiac problems, the drug has to be taken lifelong. Under such conditions, what are the other side effects that are seen will be recorded, and this post market surveillance actually gives that kind of safety and efficacy on long term usage by the patients, which you call it as phase 4 clinical trials. So, once all these are done, the drug will be there. But in this entire drug discovery process that we have discussed so far, in terms of the identifying the target, the bioassay process, and identifying the hit and lead molecule, in vitro and in vivo studies, and preclinical development of the drug and clinical development, then the drug is in the market. And there are a number of other challenges and opportunities are there in the drug process. So, the modern drug research carried out by multidisciplinary team of the chemists, pharmacologists and clinicians is not much older than a century. So, the drugs developed so far have been targeting only 500 different disease like genes or proteins, but the technological breakthroughs that have happened through genomics and proteomics, metabolomics, it is anticipated that there are 5,000 relevant new targets will be possibly there for exploitation or for developing drugs in the next 10 years. So, with emerging infectious diseases, increasing age associated disorders and need for personalized medicine, there is a greater potential for novel drug discovery and development in the years to come. And in this process, so you see enormous opportunities and potential for basic scientists to get involved in the discovery process. Discovery in the process, the most important, as I mentioned, is understanding the disease process. Once you understand the disease process, then you come up with the specific target and use this target for developing. So, in order to explain this entire disease process, as a typical example, we have taken cancer as a typical example of the entire drug discovery process. How the understanding of the disease process of the cancer in terms of hallmarks that are associated with the cancer, the signaling pathways that are involved in the thing, how these hallmarks and signaling pathways have been exploited to develop anti-cancer drugs has been explained in this course. So, when you are taking this course, you will not only understand the entire drug discovery process that is associated, but also see as a typical example like cancer, how a target has been identified, how the target, a bioassay has been developed, and how that has been exploited to develop a hit, lead molecule, preclinical and clinical development. As I said in the beginning, the expert faculty includes the basic scientists from universities, the industrialists who are involved in the drug discovery process, the R&D laboratories in various companies like Genentech, Merck, Roche, and a number of other, and in addition clinicians who are involved in the clinical drug trials. So, they were all involved in offering this entire course on cancer drug discovery and development. Hope this course will give an overview of drug discovery and development for the students in biology, chemistry and pharmacy at postgraduate, or undergraduate, postgraduate, or at PhD level and postdocs. So, hope the students get enlightened with the entire course on cancer drug discovery and development. Thank you.